So a new paper came out showing another evidence that spike protein can enter a nucleus. We discussed this in the past, showing previous evidence from one published paper and from Pfizer data itself, but this paper was very interesting in that it actually showed for the first time evidence that spike protein is directly entering inside the nucleus. My name is Dr. Mikola Rashek of Genomics, and let's get started on this topic. So as I mentioned, I've already made a couple videos discussing the possibility that spike protein could be getting inside the nucleus, but one argument against that type of information that I've discussed in the past was the fact that the images that were presented showed spike protein overlaid over the nucleus, and that does not necessarily tell us whether the spike protein is truly inside the nucleus or whether it could be on top of the nuclear envelope. Now, in this particular publication, the authors wanted to finally resolve that. Now, before I continue, I wanted to let you know this is a preprint. What that means is that this particular study has not yet been peer-reviewed, which means potentially it might not even survive peer review and might be considered inaccurate information. However, personally, I love preprints because it gives us another body of evidence that sometimes we would never otherwise have access to, not just because of the fact that sometimes preprints don't get published because they're not correct information. Sometimes they don't get published simply because they don't fit the desired narrative of the scientific publications to which the authors are submitting the preprint for such peer review. So, and preprints are now also being heavily referenced as well, but whenever you hear a preprint, always consider that has not gone through the peer review, and therefore the level of quality is not as high as what you would expect from a peer-reviewed publication. So please keep that in mind, that there's always a potential level of inaccuracy involved as a consequence of that, okay? But let's discuss what the authors did, because this was really interesting. So one thing is that the authors wanted to discuss is the fact that spike protein, in comparison of the SARS-CoV-2 virus, in comparison to SARS-CoV, the first one from over 20 years ago, it has inserts that are we have a visitor here. Sorry about that, but that's now part of the video. So it has 4 inserts, and I discussed those 4 inserts in relation to a very controversial paper that has been published, which has been withdrawn, where the authors mentioned these 4 inserts, but they claim that those 4 particular inserts come from HIV virus only, and because of that, those authors claimed at the time that this suggested that spike protein might have been manipulated to have those inserts, and that's why they faced that criticism and why the preprint itself was withdrawn, because since then it was clearly demonstrated that it's not the case. These inserts have been shown to be present in other viruses as well, including the most closest related coronavirus to the SARS-CoV-2. 3 of those inserts are also present in that coronavirus. So that's one of the reasons why that paper has faced such criticism, and just wanted to let you know that that's definitely not the case. These inserts have been observed in other viruses as well, including the one of particular interest to us that we're discussing today, which is, believe it or not, an insert that happens to, as a consequence of its existence, create the furin cleavage site. Now, furin cleavage site is very controversial. We've made, for a variety of reasons, we've discussed that in a series of our videos already as to why it's controversial. And if that wasn't enough, we're discovering yet another unusual feature of that insert. Now, remember, furin cleavage, that the presence of that furin cleavage has made SARS-CoV-2 so much more infectious to humans, and why that virus has become so much more infectious. Now it turns out that insert that created completed furin cleavage site also accidentally might have created what is referred to as nuclear localization signal, which is a type of protein signal that can be recognized by other proteins to grab the protein and bring it inside the nucleus or to the nucleus region, such as on its surface. Now, there's multiple types of these nuclear localization signals, and this one appears to be potentially one of them. So the authors who were studying this information wanted to see, well, can this truly act as a nuclear localization signal, and therefore can the spike protein enter the nucleus? So they did the same type of work like we've already discussed in a previous video. So they clearly were able to demonstrate that the spike protein can overlay its presence within the cell, overlay itself over the nucleus. Now, by the way, the cells they used were epithelial cells that were, this is not done in an animal model. It's not done, it's done in cells in a laboratory setting, in like a petri dish type of thing, if you will. And those epithelial cells, they use specialized epithelial cells constructed to mimic what would be the type of cells that are used inside our lungs. So they're actually used as a model to study potential behavior of lung epithelial cells. And those cells were the authors infected with the SARS-CoV-2 virus, and they were able to show that subsequently the spike protein was overlaying the nucleus, where the nucleus was. And then what they also did, and this is really interesting, they also look at the mRNA of the spike protein distribution, and it turned out that it also overlaid where the nucleus is. So that was really interesting and a new finding. And then they wanted to check whether that is truly inside the nucleus or not. Now, just to back up a little bit, just remember that SARS-CoV-2 virus, when it enters our cells, it provides a genome, and that part of that genome can then be used as a template to produce different mRNAs, and that includes production of mRNA for the spike protein. So this is the mRNA they were looking at, okay? And so they wanted to see, okay, is this mRNA and the spike protein truly inside the nucleus or not? So they use different technologies, high resolution imagery, and they use this type of computational software that would allow them to look at three-dimensional distribution of both the mRNA as well as the spike protein. And in this way, these are really, really neat images. I rarely get to see them, so they're always fun to look at. And they were able to show that indeed both the mRNA as well as the spike protein using this type of analysis appear to be inside the nucleus. On top of that, so this is definitely the big revelation, is that mRNA also can be found inside the nucleus. So it's the first probably evidence we see clearly showing that spike protein can be found inside the nucleus, and on top of that they are also showing that mRNA can be found inside the nucleus. They quantified that, and they were able to show that for the mRNA, 90% of the mRNA was in the cytosol, which is the liquid component inside the cell. And so 90% of the mRNA was in that cytosol, distributed throughout the cell. Approximately 10% was on top of the nucleus, and about 1% of the spike protein mRNA would enter the nucleus. Now they did that also for the spike protein, and the distribution was a little bit different. 75% was spread throughout the cell in the cytosol, and that also included spike protein entering the outside membrane of the cell. Unfortunately, they didn't quantify how much of the spike protein actually goes to the membrane itself. Too bad. That would have been really interesting information as well. I believe it was 15% of the spike protein would have resided on top of the nucleus, and then about 10% of the spike protein was inside the nucleus. And then they were able to show as well, using this type of technology and three-dimensional imaging, that the mRNA inside the nucleus always colocalized with the presence of the spike protein. So the authors actually suspect that spike protein is necessary to bring that mRNA inside the nucleus. So somehow, and it's not known how yet, it appears that potentially spike protein might have a binding site for its own mRNA and be able to bring it inside the nucleus along with itself. So what are the potential consequences of that? We don't know. We cannot know that at the moment, but definitely we should be investigating this further just to confirm whether, first of all, this is true. Yes, then like as I mentioned, this is now my third video on this topic showing potential evidence of this happening, but of course we need further confirmation. And then if it's true, what are the molecular consequences of that? There has been some suggestions made, such as potential interference with the repair of the genetic material, but again, we need much further much more analysis than what we have available to us so far. So, and also potential consequences, even authors mentions, like what does it mean for the vaccines? Because perhaps if this were to happen with vaccines as well, it would mean that vaccines might not be able, both the mRNA as well as the spike protein might not be available to the same degree to be able to stimulate our immune system. Of course, vaccines obviously do stimulate our immune system, but it means that it might limit the amount of material available to stimulate the immune system. So that also potentially should be investigated further if this were to be true with the vaccine material as well. But clearly, very interesting study. You really need imagery, and what does it mean in the future? We need to find out. So I'm gonna end there. Just wanted to let you know that we have another COVID Q&A coming up. So the first 10 people who subscribe to our newsletter, will send you free tickets to that. It was a lot of fun. There's a Q&A session. And if you're already a subscriber and you've never got a ticket, please email us. If we have free tickets available, we'll send you one. We have another event coming up also for business owners. This is specifically a program, well-being program for the employees of businesses, where me and two other experts get together to provide information on well-being, structuring well-being from different arenas of life. We're talking about financial well-being, mental health well-being, as well as physical well-being, which is what I take care of, but from the point of view of how you can use genetics to protect your physical well-being. So definitely unusual combination of different expertises. I also wanted to let you know, please check out my new series on how you can train your optimism. And the reason why is because why I'm so interested in this is because this is how you could be protecting your immune system, because there's appears to be a strong correlation in positive biological outcomes, including quality of immune system, in relation to the level of optimism. And this is the reason why I'm so interested in this, is because this is a non-invasive way of potentially improving your immune system, as opposed to any invasive way, such as including taking supplements. So please check it out, because we're only talking about doing mental exercises, so retraining your mind. And I think we do need to attempt to protect our immune system to the highest degree possible, because clearly, obviously, we have been dealing with a lot of unusual events, such as the pandemic, right? So please check it out. That's one of the interesting ways of how we could be improving our immune systems without really doing anything else except doing mental exercises. And then finally, I wanted to remind you, we have the ongoing survey for your potential interest in epigenetic testing. That's DNA testing that allows to monitor your biological aging, which can be altered based on your lifestyle choices. So please check that out. We're still trying to assess the interest in that type of service, including potentially you could be using this to determine your predisposition to negative health outcomes if your biological aging is changing too rapidly in relation to your chronological age, or vice versa. If you're not growing as old biologically as you are chronologically, that's in your favor at that point, and it's amenable. You can change it with your lifestyle choices. So please check it out as well. Once again, thank you to everyone who is sharing our videos. Thank you to everyone who's subscribing. Please continue to do so. This is how we grow, obviously. Thanks for all the comments. I try to get through some of them. Impossible to get through all of them because there's now thousands of these, but I do try to poke into that territory, and it's a lot of pleasure. So I wanted to say thank you for that. It's fun to be able to see many of you come back and leave comments, and it's a pleasure to be able to interact with with what feels like a very, very large family. So I appreciate that, and finally thank you to all of you who has been sending super thanks as well. And just to conclude, we are in a beautiful near Nordegg, Alberta, and what you see behind me is a beautiful Abraham Lake. If you're wondering why it has such a bizarre color, it's because it's glacier water. So bye everyone, and see you next time. Ciao.